The topic is plant cell suspension culture. Suspension culture refers to the growth of cells or tissues in liquid nutrient media containing growth hormones. The media is constantly agitated at a medium speed. It allows the uniform distribution of nutrients and aeration to the cell or cell aggregates. It also breaks the cell aggregates into small groups and removes any polarity of the cells. Steps involved to start a suspension culture. Any part of the donor plant can be used as a explant. The explant is excised from the donor plant and surface sterilized. The surface sterilized explant is then inoculated into a standardized auxin and cytokinin media to induce callus formation. The callus is subcultured to increase its mass. Then the callus is transferred into the nutrient media with the same composition under aseptic conditions. A friable callus is preferred for suspension culture. The suspension media is then kept for incubation under continuous agitation at a low to moderate speed. After eliminating large callus pieces, only single and small aggregates are then again transferred into fresh media to get a suspension of actively dividing cells. This suspension is then propagated by regular subculture of an aliquot to fresh media. An ideal suspension media contains a homogenized solution of only single cells, which are physiologically and biochemically uniform. The figure here shows a diagrammatic representation for starting a suspension culture. In some cases, the explant is directly inoculated into a suspension media to start the suspension culture. However, the main disadvantage of this method is the probability of high contamination. So, in most cases, the suspension culture is started with a callus. Figure A shows the callus culture. Figure B shows the microscopic view of suspension culture formed from the calli. Here, single cells and aggregates of cells can be clearly seen. Auxins at relatively high concentrations and cytokinins at low concentrations increase cell dispersion. The high concentrations of auxins maintain the cells in an undifferentiated state. The growth kinetics in plant cell suspension culture. Increase in cell number and mass can be defined as growth. The suspension batch culture shows a sigmoid growth graph. The cells go through four phases: lag phase, the log or the exponential phase, the stationary phase, and the declining or the death phase. After inoculation of cells into the suspension media, the cells first undergo the lag phase. Here, the cell number is constant. The cell tries to adjust itself to the new environment. This is followed by the exponential phase or the log phase. In this particular phase, the cells divide and multiply rapidly, utilizing the nutrients present in the suspension media. The third phase is the stationary phase. As the name suggests, the cells undergo a static state. The growth decreases. Next is the declining phase, where the cells stop dividing and there is decrease in the number of cells due to death of the cells. This is due to depletion of nutrients in the media and some toxic cellular waste that are released into the media. The suspension culture should be checked regularly for contamination. The indicators of contamination of suspension culture are change in color of the media, turbidity in the media, change in pH, change in the granular cellular appearance of the suspension culture, the smell. In some cases, the suspension culture is pipetted out and seen through the microscope to view the presence or absence of microbes. If contamination is found, the suspension culture is immediately discarded. The methods to evaluate the growth kinetics in a plant cell tissue culture are by estimating the cell weight, both wet and dry, settled cell volume, packed cell volume, cell counting, the culture optical density, the residual electrical conductivity, and pH measurements. How to estimate the fresh cell weight? A known volume of the suspension culture is drawn out under aseptic condition. The filtered cell suspension is weighed on a pre-weighed Petri plate. The difference between the final and the initial weight gives the fresh cell weight. For dry weight estimation, a known volume of the filtered suspension cell is pipetted into a pre-weighed Petri plate and incubated at 60 degrees centigrade for 24 to 48 hours in a preheated oven. The samples are then cooled in a desiccator and weighed. The difference between the final weight and the initial weight gives the dry weight of the sample. The samples are pooled every week to study the growth of suspension culture. To calculate the settled cell volume, a known volume of the suspension culture is allowed to sediment for 30 minutes in a graduated or a measuring cylinder. The sedimentation volume is then noted. Every 30 minutes, the reading is taken and noted till the difference between the two consecutive readings are less than 5% difference. To estimate the packed cell volume, the known volume of the suspension culture is pulled out under aseptic conditions and centrifuged. The sediment is volume is noted. This is again followed 2-3 times till the difference between the two consecutive readings of packed cell volume is less than 5%. The cell density of a suspension culture can be estimated by counting the number of cells per ml. The first thing to be noted is the cell suspension contains single cells rather than aggregates of cell. The aggregates of cell in the suspension culture can be removed or disintegrated by two methods. The first method is incubating the suspension culture pulled out with 8% of chromic trioxide for 15 minutes at 70 degrees centigrade. This is followed by vigorous vortexing of the mixture for 15 minutes. The main disadvantage of this method is the cells lose its viability. An alternative method that is followed for disintegration of cell aggregates is by incubating the suspension in hydrolytic enzymes as cellulase and pectinase for 30 minutes at 25 degrees centigrade with slight agitation. Once we get single cells in the suspension media, the cell counting is done under microscope using a cell counting chamber. The viability of the cells can also be checked by using dyes like trypan blue, Evan blue, methylene blue. The optical density of the culture media increases with cell concentration. Certain probes are used to actually calculate or estimate the optical density of the suspension media to estimate the growth kinetics. Another method used is to check the electrical conductivity. It is a simple, rapid, and reliable method, and there's no need of pipetting or taking out cell culture. It can be done continuously in situ or online monitoring of cell growth. Here too, the probes are used to check the electrical conductivity. The electrical conductivity in the media decreases with increase in cell concentration. It is due to the consumption of the salts and nutrients in the media by the cells. This method is independent of cell aggregation, growth morphology, and viscosity of the media. Major advantage is no sampling is required. Another method is to continuously check the pH of the suspension media. The pH of the media reduces during culture period. Certain probes are utilized to check the pH of the media at frequent intervals. The growth rate of the suspension culture can be measured by calculating the growth index and the specific growth rate. Growth index is the ratio of accumulated biomass to initial biomass. The accumulated biomass is the difference between the final and the initial fresh or dry weight of the suspension. The specific growth rate refers to the rate of increase of biomass of a cell population per unit of biomass concentration. We won't be going into detail of this. Types of suspension culture. Depending on whether the suspension culture is a closed system or an open system, it can be divided into two types, the batch culture and the continuous culture. The batch culture is again divided into slowly rotating batch cultures, shake batch cultures, spinning, and stirred batch cultures. While continuous cultures are divided into two types, the thermostats and the turbidostats continuous suspension culture. A batch culture is a closed culture system where the suspension culture has a limited amount of nutrients and cells grow in a finite volume of nutrient agitated medium. The cultures are maintained by continuously subculturing. Here, the cells follow the sigmoid curve. In a continuous suspension culture, it's an open type culture where the culture is replenished with fresh media at regular intervals and the used up media, the accumulated cells and waste products are removed. Here, the cells always remain in a steady growth phase. The figure here shows a diagrammatic representation of a batch culture and a continuous culture. In the batch culture, the media is limited and it's a closed system. In case of a continuous culture, you can notice an inlet and an outlet on your left and right side of the culture vessel respectively. Fresh media is put through the inlet and the used up media and the aggregated cells are pulled out from the outlet of the suspension culture. Slowly rotating type batch cultures. Here, the single cells or cell aggregates are grown in a specifically designed flask known as nipple flask. Each nipple flask is designed in such a way that it possesses specific number of protrusions or projections known as nipples. The explant is inoculated into these nipples. The capacity of each flask is 250 ml and the flasks are loaded in a circular manner on a large flat disc on a vertical shaker. When the disc rotates, the cells within each nipple of the flask are alternatively bathed in culture media and exposed to air. The shaker type batch cultures. These are the most common type of cultures that we see in a lab. Here, single cells or aggregate of cells are inoculated in a fixed volume of nutrient media in the conical flask and made to rotate in an orbital platform shaker. Spinning cultures. In this type of batch cultures, Large volumes of cell suspensions are cultured. The cultures are rotated in a culture spinner at 120 rpm at an angle of 45 degree. The figure on your right bottom page shows a spinning culture apparatus. In stirred batch cultures, internal magnetic stirrers are used to agitate the culture medium safely. The magnetic stirrer revolves at 200 to 600 rpm and culture vessels up to 5 to 10 liters can be used. Chemostat cultures. These are open type suspension cultures. Here, the culture vessels possess inlet and an outlet for introduction and removal of nutrient media and cells and also for aeration. The figure on your right shows a chemostat culture. The fresh sterile medium is pumped at constant rate from the inlet and equal volume of spent or old medium and cells are displaced. A steady state condition for the density, growth rate, chemical composition and metabolic activity of the cells remain constant. The figure here shows a chemostat continuous stirred culture. Turbidostat cultures. These are continuous suspension cultures where the changes in turbidity of the culture medium is measured by changes of optical density of the medium. Probes are used to check the turbidity of the culture medium. In turbidostat, an automatic monitoring unit is connected with the culture vessel and such a unit adjusts the medium flow to maintain the optical density and pH at the preset value. Plant cell suspension cultures have several advantages over intact plant cell. In suspension cultures, easy scale up is possible. One can have absolute control of the physical environment of the suspension culture. Suspension culture can also be used for large-scale production of plantlets or micropropagation. It is a best method to understand the growth kinetics of plant cell system. It helps in modeling and predicting the plant cell culture for secondary metabolite synthesis. Downstreaming of secondary metabolite synthesis to extract valuable components or bioactive compounds is easy in case of suspension culture. The main disadvantage of suspension cultures are the cells may sometimes lose differentiated characters. The rate of mutation is more in suspension culture compared to intact cell culture. Third, the growth kinetics of the cell not steady, especially in case of batch cultures. Applications of plant suspension culture. Suspension culture is widely used in production of secondary metabolites and bioactives of commercial interest in pharmaceutical and food industry. The suspension culture has single cells Hence, it is a best source of for protoplast isolation. The single cells in the suspension culture can be manipulated by the use of plant growth hormones and additives to form large number of plantlets or somatic embryos in a very short span. Suspension cultures can be used as models to study growth kinetics and cell signaling. Thank you.